Hello students, welcome to EPG Pathshala. I am Fawzia Zakir from Department of Pharmaceutics, Jamia Hamdard. Today we are going to discuss about the module Nose to Brain Delivery of Drugs with the help of case study Insulin-like Growth Factor from paper Novel Delivery Systems 1. So students, let us see what we are going to learn in this module. First we will start with brief introduction on CNS therapeutics. Then we will discuss the factors affecting nasal absorption. We will then discuss the advantages and limitations of nose to brain delivery. We will then move on to the mechanism of nose to brain delivery. Finally, we will discuss the use of insulin growth factor in nose to brain delivery. Drugs for which main site of action is brain must be localized effectively and selectively to the brain. Disease of brain such as epilepsy, schizophrenia, meningitis, migraine, Parkinson's and Alzheimer's disease require delivery of the drug to the brain for treatment. Brain is protected by blood-brain barrier and blood cerebrospinal fluid barrier. These barriers are provided with various efflect transporters like P-glycoprotein, multi-drug resistance associated protein, and breast cancer resistance protein, which minimizes the influx of drug into the brain, thus minimizing the therapeutic efficacy of the drug when given through different routes other than the nasal route. Intranasal route is a non-invasive technique to deliver drug to the brain because the drug is in direct contact with the nasal mucosa and is rapidly absorbed bypassing the blood-brain barrier and blood cerebrospinal fluid barrier. Major pathway for the drug absorption in the brain is through the olfactory and trigeminal neuron as shown in the figure. These routes are only externally exposed portions of the CNS and therefore represent the most direct method of non-invasive entry into the brain. However, the quantities of drug administered nasally that have been shown to be transported directly from nose to brain are very low, normally less than 0.1%. Although a fraction of drug is also absorbed in the systemic circulation through nasal mucosa, which is richly supplied with the blood vessel, which then reaches the brain by crossing blood-brain barrier. Nasal delivery of drugs for the treatment of brain disease is still under its infancy due to its limitations. Various research papers have shown that there is 1.8 to 2 times increase in drug transport efficiency and drug transport percentage when intranasal route is compared to intravenous route, showing that this route can still be preferred for drug administration. Nose to brain delivery has various disadvantages. For example, mucociliary clearance from the nasal cavity, which will result in efflux of drug from the nasal region. Nasal congestion can block the way of drugs transport to brain. High molecular weight polar drugs are less permeable through the nasal mucosa. Systemic absorption of drug due to richly supplied blood vessel in the nasal mucosa. Frequent use of this route can cause infection or mucosal damage. Nasal mucosa is very sensitive. Use of permeation enhancers can also damage the mucosal surface. Drug dose due to improper technique of drug administration. Irritation of nasal mucosa can cause sneezing, which can cause drug elimination out of nasal mucosa. Drug transport mechanism is still not clear. The diagram in front of you shows the mechanism of drug transport from nose to brain. It can be seen that the drug passes through the nasal vestibule, through the trigeminal nerve to the olfactory bulb or the non-olfactory regions. When the drug passes through the olfactory bulb, it has to pass through the various rate-limiting membranes of the nasal cavity, either by paracellular pathway or transcellular pathway. After the formulation, fate of drug transport. After the formulation is administered, it gets deposited on the nasal mucosa and then its absorption through nasal mucosa occurs by taking 3 different routes: trigeminal neuron, olfactory neuron, and systemic absorption. Except systemic absorption, the other two routes can directly take the drug to the brain, circumventing blood-brain barrier, while systemic absorption of drug through nasal mucosa have to cross blood-brain barrier and blood cerebrospinal fluid barrier before it is transported to the brain. Fraction of the drug which reaches to the systemic circulation after intranasal administration may be eliminated by glomerular filtration, or it is biodistributed to different organs. The biodistribution to different organs is shown in the figure in front of you. Advantages of nasal drug delivery. Nasal delivery is safe, effective, and non-invasive method of delivering the drug directly to the brain. There is no peripheral drug distribution, so there is minimal systemic side effects. Rapid drug absorption is possible through highly vascularized mucosa. Large nasal mucosal surface is available for dose absorption. There is avoidance of hepatic first pass effect, which can ultimately lead to improved bioavailability. Wide pH range of gastrointestinal tract can cause drug degradation, which is avoided in nasal administration. Various barriers in case of oral administration, like gastrointestinal barrier, blood-brain barrier, and blood cerebrospinal fluid barrier, is avoided. Because the nasal mucosa is near the brain, cerebrospinal fluid drug concentrations can exceed plasma concentrations. Intranasal administration may rapidly achieve therapeutic brain and spinal cord drug concentrations. Self-medication is possible, so there is better patient compliance. Drugs which cannot be absorbed orally may be delivered to the systemic circulation through nasal drug delivery system. Nasal drug delivery is an alternative route for parenteral drug administration. The bioavailability of larger drug molecules administered by intranasal route can be enhanced by means of absorption enhancers or other approaches as discussed previously. Slide number 10. Various factors which affect nasal drug absorption are: non-polar low molecular weight drugs can easily cross the nasal mucosa, but large molecular weight polar drugs find it difficult to cross the nasal mucosa. Pro-drug approach can increase the nasal absorption of drug. For example, conversion of drug into its salt or ester form can increase the rate of absorption of drug. Absorption decreases significantly if the molecular weight is greater than 1000 Daltons, except with the use of absorption enhancers. Shape is also important. Linear molecules have lower absorption than cyclic shaped molecules. Both pH and pKa of the drug are also important factors. pH of nasal secretion is around 4.5 to 6.5, so drug formulation under this pH range needs to be formulated to decrease nasal irritation. pKa is also considered so that the drug remains in unionized form during absorption. Volume of nasal formulation to be delivered should be below 200 microliter due to small size of nasal cavity, as greater than 200 microliter formulation will drain out of the nasal cavity. Nasal secretion can alter the pH of administered formulation, as very small amount, that is below 200 microliter, is administered through nasal route. pH change can cause the ionization of the drug molecule, so adequate buffer capacity is required to be maintained to maintain the pH of the formulation. Hypertonic solution causes cell shrinkage, and it can also affect the activity of cilia and thus drug absorption. If partition coefficient of the drug is high, then its log p value will be high, and it has been seen that there is a direct relationship between nasal permeation and log P value of drugs. Also, the site where drug is deposited and area exposed affects drug absorption. Drug metabolism in nasal mucosa due to CYP450 enzyme and P-glycoprotein efflux transporter present at the surface of nasal mucosa further limits the drug absorption. Several enzymes that are present in the nasal mucosa, for example, proteases and peptidases, affect the absorption of proteins and peptides at the mucosal membrane. The level of aminopeptidase present is much lower than that in the gastrointestinal tract. Peptides may also form complexes with immunoglobulins in the nasal cavity, leading to an increase in the molecular weight and a reduction of permeability. Intranasal pathologies may affect the nasal mucociliary transport process or capacity for nasal absorption. Increase in the viscosity of the formulation can enhance the therapeutic efficacy of the formulation. Hydroxypropyl cellulose has been used in a study as the viscosity building agent, which can effectively enhance the absorption of low molecular weight drug. But this study did not show the absorption enhancement effect for high molecular weight peptides. Mucin is a protein which is present in mucus. It binds with the drug and slows the diffusion rate. Mucociliary clearance, apart from mucin, decreases the residence time of drug in nose. Factors affecting absorption and drug permeation have been depicted in the figure. The various factors are physicochemical factors can include molecular weight, lipophilicity, partition coefficient, dissociation constant, solubility and dissolution rate, stability and polymorphism. Formulation factors such as pH, osmolarity, mucosal irritancy, viscosity, pharmaceutical excipients, pharmaceutical dosage forms, drug distribution, physical form of formulation, drug concentration, dose and volume of administration, and finally, the area of nasal membrane exposed. Biological factors involve nasal blood flow, mucociliary clearance, enzymatic degradation, transporters and efflux systems, physical condition of nasal mucosa, site of deposition of formulation in nose. Delivery device related factors include particle size of powders and pattern of deposition. If the drug is not distributed properly in nasal mucosa after its administration, then its absorption will be affected. Deposition of the formulation in the anterior portion of the nose provides a longer nasal residence time. The anterior portion of the nose is an area of low permeability, while posterior portion of the nose, where the permeability is generally higher, provides shorter residence time. Mode of administration also affects the distribution of drug in the nasal cavity. Nasal mucosal membrane is very rich in vasculature and plays a vital role in the thermal regulation and humidification of the inhaled air. The blood flow and therefore the drug absorption will depend upon the vasoconstriction and vasodilation of the blood vessels. The size, shape, density, hygroscopicity of particles and pathological condition of nasal mucosa decides the deposition of particles, while particle size distribution determines the site of particle deposition. Nasal deposition of particles is dependent on resistance to airflow provided by nasal mucosa of an individual. With breathing particles in aerodynamic range of 10 to 20 microns are deposited on the nasal mucosa. Deposition of particle on nasal mucosa is dependent on three mechanisms: inertia, sedimentation, and diffusion. According to inertia, particle size greater than 50 micron does not enter into nasal cavity. It is empirical that we discuss the various barriers for nasal drug absorption. The first barrier is the enzyme. Although nasal delivery avoids the hepatic first pass effect, the enzymatic barrier of the nasal mucosa creates a pseudo first pass effect. The xenobiotic metabolic activity in the nasal epithelium has been investigated in several species, including humans. Drug metabolism in nasal mucosa due to CYP450 enzyme and P-glycoprotein efflux transporter present at the surface of nasal mucosa further limits the drug absorption. In the phase 1 studies, cytochrome P450 enzymes have been studied extensively for their toxicological significance. Since these enzymes metabolize inhaled pollutants into reactive metabolites which may induce nasal tumors. The cytochrome P450 activity in the olfactory region of the nasal epithelium is higher even than in the liver, mainly because of a 3 to 4-fold higher NADPH cytochrome P450 reductase content. The phase 2 activity has also been found in the nasal epithelium. Several enzymes that are present in the nasal mucosa, for example, proteases and aminopeptidases, degrade the proteins and peptides at the mucosal membrane. The level of aminopeptidase present is much lower than that in the gastrointestinal tract. Peptides may also form complexes with immunoglobulins in the nasal cavity, leading to an increase in the molecular weight and a reduction of permeability. Mucociliary clearance is another barrier to nasal drug absorption. Intranasal pathologies may affect the nasal mucociliary transport process or capacity for nasal absorption. Increase in the viscosity of the formulation can enhance the therapeutic efficacy of the formulation. Hydroxypropyl cellulose has been used in a study as the viscosity gelling agent, which effectively enhances the absorption of low molecular weight drug but did not show this absorption enhancement activity for higher molecular weight peptides. Protective barriers also affect nasal drug absorption. Mucin, which is a protein present in mucus, binds with the drug and slows the diffusion rate of drug. Mucociliary clearance apart from mucin decreases the residence time of drug in nose. Intranasal nose to brain drug delivery. Intranasal nose to brain drug delivery involves three mechanisms. The first mechanism is intracellular transport mediated route, and the other two mechanisms is extracellular transport mediated route. Intranasal nose to brain drug delivery. Intranasal nose to brain delivery involves two mechanisms. The first one is intracellular transport mediated route, while the second mechanism is extracellular transport mediated route. The intracellular transport mediated route involves a relatively slow process, taking hours for intranasally administered drug to reach the area of the olfactory bulb. The extracellular transport mediated route involves rapid entrance of drug into the brain, which can occur within minutes of intranasal drug administration. In the first extracellular transport mediated route, intranasally administered drug first crosses the gap between the olfactory neurons in the olfactory epithelium, which are subsequently transported into the olfactory bulb. In the second extracellular transport mediated route, intranasal administered drug may be transported along trigeminal nerve and cross the blood-brain barrier. After reaching the olfactory bulb of trigeminal region, the drug may enter into other regions of brain by diffusion, which may also be facilitated by perivascular pump, that is driven by arterial pulsation. Cellular mechanism of transport from nose to brain. Passive diffusion allows drugs to move down the concentration gradient through the nasal mucosa. In addition to passive diffusion, molecular weight and lipophilicity of the drug also affects the drug diffusion. Lipid-soluble drugs with molecular weight less than 400 Daltons can cross biological barriers. For example, progesterone, which is lipid-soluble small molecule, has cerebrospinal fluid concentration greater than plasma concentration, according to a study conducted by Jogini and his co-workers. Carrier-mediated transport allows poorly permeable drugs to cross the cell membrane. This transport mechanism is of two types: facilitated diffusion and active transport. Facilitated diffusion is a downhill transport process and allows movement of solutes through glucose transporter and equilibrative nucleoside transporters. On the other hand, receptor-mediated transport involves peptide which binds with the receptors, which then gets internalized. Peptides like transferrin, albumin, insulin growth factor, low-density lipoprotein, ceruloplasmin, etc. are transported by this mechanism. Insulin-like growth factor. Insulin-like growth factor, formerly known as somatomedin, is a peptide hormone. Its function is to stimulate growth, but it also has the ability to decrease blood glucose levels. The name insulin-like growth factor reflects the fact that these substances have insulin-like actions in some tissues, although they are far less potent than insulin in decreasing blood glucose concentrations. There are two types of insulin growth factors: insulin growth factor 1 and insulin growth factor 2. Both are similar to insulin in terms of having 50% common amino acid. But unlike insulin, which is synthesized by pancreas, these factors are synthesized by liver. Insulin-like growth factor 1 inhibit the secretion of growth hormone by pituitary, but the physiological role of insulin-like growth factor 2 is unknown. These two factors are also distinguishable in terms of specific actions on tissues because they bind to and activate different receptors. Patient deficient with growth hormone have low concentration of insulin growth factor 1 and is higher in patient showing higher level of growth hormone. Insulin growth factor 2 level is little affected with the concentration of growth hormone, indicating little effect of insulin growth factor 2 on growth. Insulin growth factor 1 had broad spectrum of biological action. It stimulates bone formation, protein synthesis, glucose uptake in muscle, neuronal, survival, and myelin synthesis. It also reverses negative nitrogen balance. For these reasons, insulin growth factor 1 has been proposed as a therapy for osteoporosis, diabetes, obesity, neuromuscular disorders, growth hormone resistance, and insulin resistance. Rationale for intranasal administration of insulin growth factor 1. Oral route is not suited for the administration of insulin growth factor 1 due to rapid degradation of insulin growth factor, and thus therapeutic concentration in blood is never reached. It has been reported that there is a possibility that insulin growth factor 1 can cross blood-brain barrier. Parenteral administration may bypass hepatic metabolism of insulin growth factor 1, but due to extensive binding with plasma protein, free insulin growth factor 1 in blood is never circulated, and thus little free insulin growth factor 1 is available to cross the blood-brain barrier. But approaches could be used to saturate plasma protein with insulin growth factor 1 by administering intravenous bolus dose of insulin growth factor 1, which causes transient rise in insulin growth factor 1, which is free to cross blood-brain barrier. But this high concentration of the hormone can result in hypoglycemia. There is some other side effect which could result from insulin growth factor 1 due to its interrelationship with growth hormone and somatostatin. In consequences result from cerebral infarction, Alzheimer's disease, and the central manifestations of amyotrophic lateral sclerosis and multiple sclerosis. There is need to deliver insulin growth factor 1 directly to target organ, that is brain in this case, and it is possible if brain is directly targeted through intranasal route. We will now discuss insulin-like growth factor with the help of a case study. The possible pathway of delivery after intranasal administration of insulin-like growth factor was studied through autoradiography. Insulin-like growth factor was labeled with iodine 125 and were given intranasally or intravenously. Male Sprague Dawley rats were selected for this study. After administration of insulin-like growth factor 1, rats were killed. Gamma counting of micro-dissected tissue and high-resolution phosphor imaging of tissue sections showed distribution of insulin-like growth factor radio-labeled through two routes. One route was olfactory bulbs and the other was trigeminal region, which directly leads the way to the CNS. Lymphatic drainage of intranasal administered compound was also seen due to its targeting in cervical lymph nodes. Intravenously administered radio-labeled insulin-like growth factor did not reach to the CNS at the same amount as that administered through intranasal route. There was similar distribution of intravenous administered insulin-like growth factor as that administered through intranasal route, as shown in the figure. The diagram in front of you shows the distribution of radio-labeled insulin growth factor 1 in blood after intravenous and intranasal administration. After intranasal delivery of insulin growth factor 1, it showed activation of the hormone signaling pathways, showing that the portion of intranasal administered insulin growth factor 1 reaches the brain. Trigeminal and olfactory are the two routes which directly takes the insulin growth factor 1 to the brain, circumventing blood-brain barrier. In another study timed, pregnant Sprague Dawley rats were used in the laboratory on day 19 gestation. Intracerebral injection of lipopolysaccharide were given, which resulted in oligodendrocyte and white matter injury. Recombinant human insulin-like growth factor were administered through intranasal infusion into neonatal rat brain. Recombinant human insulin-like growth factor attenuates lipopolysaccharide-induced apoptotic cell death. Intranasal infusion of insulin-like growth factor 1 up to 2 hours after lipopolysaccharide exposure attenuates lipopolysaccharide-induced brain injury. Intranasal infusion of insulin-like growth factor 1 improved neurobehavioral deficits induced by lipopolysaccharide in juvenile rats. Intranasal neurotrophin. Neurotrophin are known for their ability to protect and promote the growth of new neurons and makes them a good candidate for the treatment of neurodegenerative disease. But they are not able to cross the blood-brain barrier efficiently and demand invasive injection methods. It has been suggested that intranasal administration of neurotrophins gives a desirable way to target neurotrophins to the CNS to treat neurodegenerative diseases. It has been shown that in a mouse model of Alzheimer's disease, intranasally administered neurotrophins reduces neurodegeneration and improved performance in memory tasks. So students, let us summarize what we have learned in this module. We have first discussed the fate of drug delivery through nose to brain, the various factors affecting drug permeation and transport. We have discussed the rationale for delivery of insulin-like growth factor through nasal route with the help of a case study.